Let's go over the process of translation. Remember that transcription is going to occur before translation. Transcription occurs when RNA polymerase makes a copy of DNA into mRNA. Now this mRNA stands for messenger RNA, and it's going to leave the nucleus and find a ribosome. Translation is specifically the process of taking that sequence of nucleotides in the mRNA, decoding it, and making a protein. And we make proteins at ribosomes, so translation will occur at the ribosomes. Now, whenever the ribosome attaches to the mRNA, it's going to start reading that nucleotide sequence. Whenever the ribosome starts reading that sequence, it's going to come across a codon known as the start codon, which is AUG. tRNA molecules are going to bring in amino acids that's going to correspond with the codon sequence in the mRNA. For example, AUG is going to code for methionine, or MET. These tRNA molecules will also have an anticodon sequence that's complementary to the mRNA sequence. As you can see, the anticodon sequence for AUG is UAC. The ribosome will continue down the mRNA sequence, reading the codons and translating the message. Each new tRNA molecule will bring in a different amino acid that will keep bonding to the previous amino acids. Whenever the ribosome reaches the stop codon, the ribosome will detach from the mRNA, freeing the protein up to be folded later and to have some specific function in the cell. So the protein's amino acid sequence is going to be specific to its structure. And the structure of that protein will be specific to the function. And how those amino acids bond together will determine the structure and function of the protein. So the function of a protein will be determined by its structure. The structure of a protein will be determined by the amino acid sequence. The amino acid sequence will be determined by the mRNA sequence. And the mRNA sequence will be determined by the initial gene or the initial DNA.